Hello.
Hello,
Uh,
Professor. How are you?
so I, yes, I'm George. And so pleased to have a chance to connect today. This is great. Uh,
Thank you. Thank you for joining
so
us. Hello, everybody, and good evening, good morning, anywhere you are. I don't know. Some of the participants' situation might be. It's a daytime. Here is a nighttime. Tonight, you are very lucky. You have a great professor. He's George Schatz. Already, we have shared his CV with most of the participants. So I don't spend too much time introducing Professor. He's already well known. It's enough for Professor that he has more than thousands of peer-reviewed papers and countless citations, really countable. As the number of citations really is. We appreciate your work, Professor, in this field. And tonight we are listening to be. Uh, your student for a few minutes to take some experience from you. So, Professor, I already shared your CV, your short CV with the participants already. It's published on website, published in our social medias. So, if you permit, I'll give you the mic to you and proceed directly to your subject. We are hearing you, Professor. You have a mic.
yes. Okay. Yeah, are we ready to get started? Should I get started? Yeah. Okay. Let me just share the screen and then we'll get going. Oh, sharing is not turned on right now. Can you turn the sharing on?
You don't see your screen, Professor.
uh, Here. Well, that's because I need to share
Okay, okay.
my.
Proceed this time. I permit you to share your screen, please.
Oh, okay, good, got it. Okay. All right, let's do this. Okay. All right.
Yes,
Can you see
please.
my slide?
Yes,
My
yes,
first slide?
please proceed. Yes.
Okay, very good. All right. Yeah, let me just move over here so that I can be on top of this stuff. Okay. So yeah, so I'm George Schatz. I'm on the faculty in the chemistry department at Northwestern University. For those of you who don't know where Northwestern is, it's actually not in the northwestern part of the United States. It's actually very close to the city of Chicago. In fact, this is an aerial view of the university looking south, and here's the city of Chicago that's off in the distance. And this is the shore of Lake Michigan. So in the end, we're right next to Lake Michigan. So that's something we are exposed to all the time. In the summertime, it's very nice. It's very pleasant temperatures. In the wintertime, it tends to be cold and snowy, but that's okay because we enjoy the summers and in some ways the winters. This is another picture of the same campus going from the other direction, looking north. And again, you can see that we're right exactly on the shore of Lake Michigan. Okay, so that's where we're located. This is a picture up in the top of my research group, and we're standing in front of a building, which is where we're located. The building was the home previously of two people that won Nobel Prizes. Okay, so John Pople is this guy, won the Nobel Prize in the late 1990s, and Friedrich Studer is the other guy, and he won the Nobel Prize in about 10 years ago. And so in the end, this is the types of things that go on. But of course, who knows going to be who's going to be the next Nobel Prize winner? We don't know. All right. So now let me move on to so here I'm just saying a little bit about myself. I was undergraduate at a place called Kscien University, which is in New York State. And then I did a PhD at Caltech. And later on, I was a postdoc at MIT. And I've been at Northwestern for nearly 50 years. And my research has mostly focused on theory in combination with nanoscience, and you're going to see mostly the nanoscience version of it today. I decided I wasn't going to get too much of the theory. And I have, written a bunch of papers. For a long time. I was the senior editor and then the editor-in-chief of the Journal of Physical Chemistry. Okay, and so that started out as just one journal, but then by the time I stepped down from it in 2019, it was these four journals. Okay, and these are among the largest journal combination in all of science. Okay, with publishing of like 12,000 papers a year, things like that. So anyways, but I stepped down and now I'm just doing regular research. So what I'm going to do is to first tell you a little bit about something called plasma excitation, because that turns out to determine the optical properties of silver and gold nanoparticles and other kinds of particles too. And so that turns out to be what this talk is mostly about. And we'll talk about something called extinction spectroscopy and surface enhanced Raman spectroscopy, because those are both techniques that are very special for silver and gold nanoparticles. And then we'll talk about practical applications of these kinds of spectroscopy for the use of gold and silver nanoparticles. And there's going to be some mixture of medical applications and also directions that relate to things like electronic devices, things like that. All right, so this starts with this picture where we're talking about colloidal nanoparticles. Colloidal nanoparticles would be little particles like 10 nanometers size particles of gold that are suspended in water. And it turns out that makes a red-colored liquid. Okay, and so that's the people who were originally studying this material. Michael Faraday studied it in the 1850s, and he was fascinated by the fact that there was this gold color, and yet it wasn't. He wasn't sure what was responsible for the gold color. Obviously, it doesn't look like a piece of solid gold at all. Okay, and so the question is, why did it have this funny color? Okay, and he did things like take a test tube full of this colloidal material and evaporate all the water. Okay, and it turns out when he did that, he got gold. Okay, solid pieces of gold. So he knew that it was just simply something about small little particles of gold. But in his day, he couldn't divide the particles up enough to actually show how they turn into this red colored material. If you take the optical spectrum of the gold particles, you get this result right here, where there's a peak in the spectrum at about 520 nanometers. Okay, that's in the green region of the spectrum. Okay, and so it might be a little bit of a puzzle as to why this stuff looks red when that peak is definitely green. Okay, but it turns out that has to do with just what we mean by color. Okay, and so extinction, actually, this is a plot of extinction, which is the sum of absorption and scattering. Okay, and it turns out that when you look at the light that's through here, you're looking at the light that passes through the sample that is not absorbed or scattered. Okay, so in the end, that this 500 nanometers is where there's a lot of extinction. Okay, but that means that the light that you're going to see in this kind of experiment corresponds to the light that's not absorbed, and therefore that's going to be over here at 600 nanometers, which is in the red region of the spectrum. So that's why it looks red. Now it turns out that if you do the same thing with silver, so little 10 nanometer particles of silver suspended in water, you get this yellow color, and that's because this feature that corresponds to the maximum over here, the corresponding spectrum of silver has a peak at about 390 nanometers. Let's just say that. Okay, and that's now in the the blue to UV region of the spectrum. And as a result, the light that's transmitted in this case is kind of over here. So that's some combination of red and green and things like that. So you get yellow out of it. Okay, so in the end, that's a neat little story about just colors. It turns out the properties of these colors for colloidal materials have been known for a long time. And here I'm showing you a famous cup called the Lycurgus cup that you can find if you go to the British Museum in London. You can see this cup. Okay, and it turns out that that when you shine light down from the top through it, it looks reddish in nature, and that's this color over here. On the other hand, if you have shine light in from the side, then you get reflected light, so that's going to be the green light. Okay, so in the end, this is really the essence of this property that's called plasma excitation. And what that means is the following. If you think of these particles as being little spheres, and then there's going to be the nuclei and then the electrons. And it turns out that when you shine light on them, you're applying an electric field to the spheres. And what that does is it moves the electrons, but not the nuclei. Okay, and so what happens when you have plasma excitation, then it turns out what you're doing is taking this cloud of electrons and you're displacing it relative to the nuclei. And it turns out there's a restoring force such that if you just let the electrons go, then all of a sudden they start oscillating back and forth with some frequency. And this oscillation of the collective oxidation of the electrons is called plasma excitation. Okay, and there's a characteristic frequency that arises for different metals. And it turns out for gold, it's just 520 nanometers, and for silver, it's 390. Okay, so in the end, every other metals have other plasma frequencies, but that's what it is for these two metals. Okay, so that's the basic idea that has been known actually for a long time. Again, Michael Faraday studied it. Of course, the stained glass windows and this Lycurgus cup go back to Roman times, and of course, even before that by a lot. And so in the end, in some respects, this is not really something new. But nevertheless, there's been a lot of changes in this field in the last 30 years. And so that's really what I'm going to focus on mostly in terms of where this is going to go. Now, there's something called Rayleigh scattering. Rayleigh scattering is also, it's light scattering. Okay, it is light scattering where the frequency of the light is not changed by the scattering off of whatever you're scattering off of. Turns out if you take colloidal silver, and so just think of a solution that contains a whole bunch of silver particles that looks like this. Okay, and then you do this experiment where you have white light coming in from the side and hits these particles, and then you can get scattering, say at 90 degrees, and this scattering is what you're seeing in this picture. So in this case, it was done in a microscope. There's an apparatus called dark field microscope that scatters the light and through a microscope so that you can focus on the light coming from each individual particle in the solution. So there's a lot of particles here, and each one scatters. And it turns out in this case, I told you that the plasma in silver was 400 nanometers. So you might imagine the scattering might occur in the blue region of the spectrum. And there's a lot of it that does scatter in the blue region of the spectrum. But it turns out that some of these particles are not spheres. Some of them are other shapes of particles. And so that ends up leading to light scattering in the red. And here's the yellow ones over here. Okay, green, all throughout the visible region of the spectrum. Okay, so this is again, it's just due to plasma excitation, but it also tells you then that this plasma wavelength depends on the size, on the shape of the particles. It also depends on the size. Here's maybe a more quantitative story related to that. I used to have a guy in my research lab here, Rick Van Dyne, and he did this experiment where he made little, they're gold colors, but they're really silver. They're made out of silver nanoparticles. Okay, and they're shaped like a triangle because you're vapor depositing the silver atoms through a mask that looks like a triangle. So in the end, you get these triangular shaped particles, and the experiment he was doing was such that he could vary the length of the edge of the triangles. He could also vary the thickness of the triangles. Okay, and so in the end, remember I said that silver has a plasma at 400, but that's if it's a sphere. Okay, and so it turns out that if you make it some other shape, like a triangle, and then you vary the shape of the triangle a little bit, then you can tune this plasma through the visible region of the spectrum all the way 800 nanometers here. Okay, is in the near infrared, and you can even go to the further to the infrared from this. And so again, it's just telling you the properties of these particles and the fact that you can change those the colors of the particles by just changing, in this example, just their shape. All right, so what if you think about the periodic table? What is the which metals are plasmonic and which ones are not? Okay, and so a guy who was a postdoc in my group came up with this idea, and what he did was to calculate some property that's actually this property that's down here, and which has to do in a sense with the the brightness of this plasma that is emitted. And it turns out if you look at silver, the number he came up with in this measure was 97 for this. It's a dimensionless number, and it's just 97. Okay, and so silver turns out to be the best plasmonic material in the entire periodic table. Gold is 34, so that's actually still very good. So there's a lot of things that we do with gold, and gold has better chemistry properties than silver. So that's why people like using gold. And then copper, it turns out, also works. Okay, it's 10. All right, so there's some things we've done with copper. Aluminum also works. That's about 14. So that's another good metal for plasmonics. Okay, and then you also see that over here, the alkali metals are also very, very good. Okay, but it turns out people generally don't do things with the alkali metals. They could, okay, but the alkali metals are particularly if you look at the ones further down the line, potassium, rubidium, cesium. Okay, these guys spontaneously combust in air. And so you sort of don't want to do anything, at least with the metallic forms of these elements. Okay, so people don't do much with those, but there's many other metals actually that are somewhat interesting. Magnesium is somewhat interesting. It's about 10, and we have done experiments with gallium and also with indium, and those are two pretty good plasmonic materials. We did do one paper once with palladium, which is good for a few properties. It's somewhat more limited. And there's a few other things. Tantalum is pretty good. Okay. And so in the end, there are a lot of opportunities. And of course, you can make alloys. You can make other materials that are not metallic, which are also plasmonic. And so in the end, it's actually a pretty rich field. But in the end, today, I'm only going to talk about silver and gold because those are the two most popular materials. All right. So this guy, Rick Van Dyne, that I was mentioning before, he did an experiment in 1977 that was astounding. Okay, so first of all, he made silver particles. These are 1970s versions of silver particles. Not too pretty, but that's what they got. And what he did is take these particles, and they were on a surface. And then what he does is he shines light on these particles, and there's molecules that are on the surface in present as a monolayer of molecules. The molecules that he studied in this example is called pyridine. The shape of this molecule is indicated right here. Okay, and what he did was to look at light scattering from the particles and the molecules. Okay, whatever you got. Okay, it turns out if you looked at elastic scattering, he would be looking at Rayleigh scattering. So that's similar to what I showed you before. But in this case, he looked at what's called Raman scattering. Raman scattering was an effect that was discovered about 1930 by a guy named Raman in India. And it turns out that what you get is a spectrum. This is the spectrum right here of pyridine in solution. And you get a spectrum that tells you about the vibrational modes of the molecule. Okay, so in other words, each vibration can couple to the light that you're exciting with and produce a new frequency that is shifted from the initial excitation by the vibrational frequency. So that's called Raman spectroscopy. And here you see a bunch of peaks in a spectrum. And it turns out those are telling you which vibrational modes of the pyridine molecule are able to interact with the light. And so this type of experiment has been done for since Raman's time. But it turns out that what he did in this experiment was to look at a monolayer of molecules on these particles. And what he got was more or less the same Raman spectrum. But it turns out that if you work out the intensity per molecule in this spectrum compared to the one where this is in solution at a 2.5 molar concentration, you discover that the intensity per molecule here is 1,000,000 times higher than it is up here. And so that was an amazing result. Anytime you do a measurement and you get something that's 1,000,000 times more intense than you expected to get, that's going to be a big deal, and it was. Uh, and so ultimately, this was explained in terms of how light, so again, remember, it's oscillating electric field and it interacts with a gold particle, and when it does, or silver particle, and in this case, it induces a dipole moment that's oscillating up and down in the particle. There's an electric field associated with that particle, and that field, I'm showing you contours of the field outside the particle here. And it turns out the molecules are right on the surface of the particle, so they see that field. And so it turns out that in some cases, because of this plasma excitation, the field that you induce here is actually larger than the field that you started with in the original excitation. And it can be even like 10 times larger. But what happens is that if the field is enhanced, then the Raman intensity is enhanced. And it can be enhanced by an amount that goes like the 4th power of the field. And so as a result, if it's a factor of 10 enhanced, you get 10 to the 4th for the enhancement factor. And in this experiment, they even got a bigger number, 10 to the 6th. Okay, so this was a remarkable discovery. And so people ever since then have been trying to figure out how to take advantage of this result. Okay, so that's great. And then what I'm going to do now is to tell you about some applications that have been done with these gold and occasionally silver particles. Okay, on a variety of different applications. I'll tell you first about how gold particles have been used in disk drives that are used in computers. Okay, so that's one. And then we'll start getting more into the biomedical direction. So how it is that you can use gold particles in combination with antibodies to detect various diseases. And one of the best examples of that is this COVID test that probably everybody is familiar with. And then we'll talk about glucose sensing using Raman spectroscopy. And we'll talk about some related things like protein sensing and other kinds of diagnostics that you can do that allow you to detect interesting molecules with Raman. And then at the end, I'll talk a little bit about tags that you're used that are in currency for identifying counterfeiting. Okay, so this application to magnetic recording media, okay, it started about 2012 or 10 or so. It turns out that this is a technology that was developed by a company called Seagate. Seagate is a disk drive company, okay, and of course, as you can imagine, over the years they've needed to shrink down the materials and all that stuff for the disk drives, and so in the end, that's what came along when they developed a method called HAMR, heat-assisted magnetic recording. Okay, and it turns out, what is that? Well, it turns out there's like in a disk drive, you have to create a magnetic field in a magnetic material, and then you also have to erase it. Okay, so this has to do with the erasing part of this magnetic signal. And the erasing is done very simply by just heating something, heat the magnetic material, and that can destroy the magnetization. And so that's the idea. And the way they developed to do that erasing was to have this little structure that's right here. Here I'm showing it from the side, and it turns out on the end of the structure is a bunch of silver, or it's actually not silver, gold nanoparticles. Okay, right here. So in the end, the way you erase is to have light that's actuated that goes to the specific portion of the disk media that you want to erase, and that light interacts with well, initially just silica, okay, which is transparent. But at the end of this is a whole bunch of gold particles, and then those gold particles heat up when you shine light on them, and that heats up the nearby material in the magnetic media, and that's what erases the signal. So it turns out that this became a big deal. People of disk drives were doing this already in commodity properties. Okay, but it turns out that since that time, the gold has mostly been replaced by well, gold alloys and even some other materials that aren't gold. But it doesn't really matter. The key thing is that you have plasmons that you can excite, and that leads to heating. And so in the end, this is an application where the plasma excitation was important. Okay, so then now what I want to do is talk about chemical and biological sensing. So this is a review article that goes back again 2012, and what they were talking about is you'd like to have a gold particle, and you'd like to make it so that somehow it will interact with some sort of analyte, and the analyte might be something that is a disease-related analyte, like an antigen for COVID. Okay, and so the idea then is that you take the gold particles, and in by 2012, people figured out how to take antibodies. That's what this Y-shaped structure is supposed to represent. To take antibodies and to chemically tether those antibodies to the gold particle. So the antibodies, of course, are recognition elements. All right, and so they can bind to the bad molecules, the antigens. Okay, and so when that happens, so imagine that you made some of this material, and then you add in the analyte, and then the analyte binds to the antibodies. All right, and ultimately, then you have to figure out how to detect that binding event. That's called signal transduction. Okay, and so that's what they understood at that point. There are some signal transductions where, for example, you can excite fluorescence, and the fluorescence in the antibody is different whether it's bound to the gold particle or not. So that's a version of how that can be done. Okay, but the version that everybody knows about where this has been done has had to do with the COVID-19 test. Okay, and so that's where we have these kind of strips. Okay, and you take a sample and you put it into this area right here. The sample mixes up with gold particles that have antibodies attached to them. And then if it has the COVID virus antigen, then it turns out you get antibody antigen binding, and then in the gold particles. And these gold particles migrate up in this direction. Okay, and eventually they get to this test strip that's right here. Okay, and it turns out the test strip ends up having additional antibodies present. And these antibodies are designed so that they will also just bind to the COVID-19 antigen. And so what that does is then that immobilizes, freezes out the motion of the gold particles. They basically just get stuck here. And so you get a red line from the aggregation of the gold particles, and that's just the red color of colloidal gold that I was talking about at the very beginning. Okay, so that's that version of it. But then it goes on to a control strip that in which the tiny particles that get through the first time get attached to antibodies in the second place. Okay, and these antibodies don't care about COVID or anything like that. They just bind directly, and so to the gold particles, and so therefore you also get another red color when that happens. All right, so that's the idea. That's the basic principle of the thing, and it's a very cheap technology. The cost of the gold particles that's used in these strips is a tiny fraction of the cost of the whole test. Okay, it's just a few cents per test. Okay, so that's that deal. All right, so yeah, and then here my colleague Van Dyne decided, oh, let me see if I can detect glucose with surface enhanced Raman spectroscopy. So here I'm showing you and some spectra that he generated. The first one is done with this model called decane thiol. It's not of interest, but just shows that you can get a Raman spectrum. And then the second one is where he added glucose to it. And what you see is that there are some extra peaks that show up in the spectrum. Okay, and so that says, it must be the glucose is somehow giving a rise to a detectable signal. Okay, and so you can take the difference between the two, and that presumably is telling about glucose. And then he did do some work with just taking the spectrum of a material that had pure glucose in it, crystalline glucose, and actually the peaks don't all line up. That's a more complicated story, but some of the peaks line up, and so that was the idea. so anyways, and then he did this where you take a sample where you know the concentration of glucose, and then he uses this method, Raman spectroscopy, to predict what the concentration ought to be. And so this is just simply prediction versus actual. And the good news is everything fell reasonably well along a straight line. And this yellow patch right here is telling you what the physiological concentration of glucose is. So the idea then was that you could use this as a method for doing a glucose test. And at least the initial goal in this thing was to do it non-invasively, so that people who have diabetes, that was where you had to take these, do these pinpricks to do a blood test. And so you'd like to be able to avoid that and just do it non-invasively. And since you're using light to irradiate the system, that maybe that could be done. Okay, so in the end, that actually does work. Okay, here's the good news: it works. But it turns out in the end, it's too expensive. Okay, and so partly that has to do with the fact that you need a laser to do the Raman spectroscopy, and you have to have a spectrometer as well. And so in the end, it's still too expensive to use this, but it does work. All right. I'm just going to skim through a couple of more examples. We did work on a similar story using Raman spectroscopy to measure the serotonin, which is a neurological agent. Okay, so this is where we had cube-shaped gold particles, and we put them onto just filter paper, and then you could put samples that contain serotonin on them, and you get a Raman spectrum. And I'm just showing you here that it matches up with the serotonin in a solid material. So this idea is a way to get to detect neurological agents. And then it turns out another colleague of mine, a guy named Chad Merkin, has done a lot of work with using the same kind of ideas, but here not looking at Raman spectroscopy, but looking at just light scattering instead. Okay, and using that as a way to detect proteins that are important in various types of cancer. Okay, so PSA is a well-known one. Okay, that he demonstrated you could detect. Okay, and so in the end, it turns out he can detect proteins. Actually, his ability to detect proteins is such that he can detect proteins that are not just the standard ones that everybody is already doing, but proteins that are in smaller concentrations. And he commercialized this technology. There's a company called Nanosphere Incorporated, although it just recently got bought up by somebody else. But this company has been around for nearly 25 years. And then finally, I was going to mention that SIRS has also been used in anti-counterfeit labels. Okay, so the idea then is you just put molecules, you embed them in, say, some sort of currency, and then the molecules would have characteristic Raman spectra. I'm showing some examples of Raman spectra that people have used for banknotes. And so that then gives you a way of identifying that the currency is what it should be and it's not a counterfeit. Okay, I tried to look up and see what currencies are currently being used by this technology. What I discovered is that nobody is willing to tell you that, okay, because they want to keep the molecules and everything else like that secret. But basically, it does work, and it does provide a way that you could screen currency for counterfeit currencies. Okay, so that's it. That's all I really wanted to say, and I appreciate having the opportunity to speak to this group, and hopefully you got at least some understanding of how the transition then from the stuff that I do, which is at the very fundamental level, into more of the practical applications of the technologies that we've been pursuing. All right, thanks very much.
Thank you so much, dear Professor, for your nice and conclusive presentation. I received several questions, but some of them, or most of them, they are repetition of the question. Most of the audience, they are eager to know how you can publish this huge amount of papers in your live research lab. What's the
Uh,
secret behind it? They're asking too much about that.
Well, partly is that you got to realize I'm an old person. Okay, so I've been doing science for over 50 years. Okay, and So yeah, so you get a lot of opportunities if you have just simply do it for a long time. The other thing is I've been extremely fortunate. Okay, I work at a major university, so I've been able to get research grants to fund a research group, and I showed you a picture of my research group at the beginning of the talk, and so therefore I've been able to get many people engaged in the science that I do, and and then the other thing is that I have lots of wonderful colleagues that are my collaborators, and so we get involved in these collaborative projects, and so as a result there's just a flow of projects coming from different directions that is happening all the time. And so yeah, no, it's really been a fantastic opportunity to be able to get involved in all these different projects. Right now on my desk, I have like a half dozen manuscripts that are in various stages of completion. Will probably get submitted in the next couple of weeks. So yeah, it's just a
Yes,
major activity.
Professor, some of the audience or some of the participants asking that for how long you are you are conducting research, in how many years you are busy with the research in your lifetime.
Oh, right. Yeah. So yes. So I got my PhD in 1976, it turns out. And so yeah, so we're just about 50 years since I received my PhD. And actually I wrote a few papers before I got my PhD. And so yeah, so that's a long time. Okay. I've just been very lucky that I've been able to maintain a high level of activity for that length of time. Then I have all these talented people that work with me, and it's not like I'm writing every single paper. I get students and postdocs to
Yeah,
uh, to to be
yes,
involved in the writing of the manuscripts. So it's
yes.
always been a really good thing.
Another repeated question. This is more than two participants asking about that. Being an editor for a journal helps you in getting citation or getting more published?
uh, well, it probably helps, so people got to know who I am, but because if they read papers in the journal, or especially if they submit papers to the journal, then they had to, they would have to write me a letter saying I want to submit this paper to the journal, and so in the end, people got to know me in terms of who I am. And so a consequence of that is that when I would publish papers myself, then they would look at it and say, oh, here's this guy who's the editor of the journal, and maybe this is an interesting paper. But so that's certainly a piece of that story. I think the other thing, though, is that there's a number of papers that I was involved where, some discovery that was reported that ended up being significant in some broader sense. Okay, so it's a little hard to go into details on it, but some of the technologies I talked about today are ones that relate to that topic, particularly ones that have to do with making the nanoparticles. Okay, because initially the colloidal particles that I was talking about were not really well defined, and they really were not the particles you want to use, especially if you're doing surface Raman spectroscopy. So there was a lot of work trying to understand how to make particles that would be better. And so usually by altering shape, size, or maybe sometimes it had to do with what were the substrates for the particle. Anyway, so I got involved in a lot of papers related to that. And those are some of my highest cited papers have to do with that.
Yes. Dear Professor, other question. Also, more than three participants ask you the same question. We are from developing countries. We have shortage in uh, infrastructure. We have shortage in grants for research.
Yeah.
What's your
Right.
advice for us? What shall we do in conducting research?
Yeah, no, that's a very good question. And I think, and I see versions of that story in many places that I visit. There's various ways to answer it. I mean, the thing in with hindsight that was really nice about some of the work that we did with these colloidal particles is that colloidal particles are inexpensive. Okay, it's just even though if there's silver and gold, it isn't very much silver and gold, and so you can make a lot of particles with tiny amounts of materials. And so that type of science was not expensive. Doing optical spectroscopy is relatively inexpensive. Doing Raman spectroscopy was a little more expensive. That was a limitation, but it has gotten cheaper over the years, and so there's more and more use of that method. Some of the basic ways to make particles are just using wet chemistry. You can also use vapor deposition, which is a way that was done in some of the stuff I talked about. And so yes, there are inexpensive ways to do science, and in many respects, there's need for more of that. Okay, and then the yeah, so then then there's other questions about what else. Yeah, so of course so in the United States there's there's a lot of high tech equipment and all that kind of stuff. And there are ways in many cases to get involved in collaborations with people that are doing something. I mean, it's usually I mean it's always hard to make connections. Sometimes there's ways to make connections in one way or another. But I think in the end it's mostly just common research interests. Okay, you start studying a topic, you find out who's doing interesting work in that topic, you try to figure out how you might contribute to that topic, and then ultimately it gets to the point, well, gee, well, you should try to uh, you know uh, contact that person. That's the amazing thing about email; it just goes anywhere, and so that turns out to be something that happens now and then. And then the other thing is the journal publications. Okay, so Journal of Physical Chemistry, they do not charge for publications. Okay, and so they do publish papers from essentially almost every country in the world. Okay, I actually did a survey on that while I was editor, and I said, are there any countries that have not published a paper or had a paper that was published in Journal of Physical Chemistry? And yeah, I mean there probably are. Okay, but I think any country with a university, with where people can do research is going to end up, leading to people that can write papers and publish in a journal like the Journal of Physical Chemistry. So that's a way to get noticed. Okay, if you publish in a journal, then people will discover your work. Okay, and that's a way to sort of gradually also get to know people. So I think there are ways to do it. It's a lot of times it is a matter of figuring out, and going to meetings, of course, is another way to get to know people. But it is a challenge. Okay, and I think we always run into this thing of just how to have influence in science, and so it can be a difficult issue to deal with.
Thank you. Professor, dear participant, we're already about 15 minutes behind our schedule. Dear
Okay.
Professor, what's your last message, especially for young researchers in the developing country? If you give us your last message, thank you so much.
Yeah, sure. Well, no, first, let me say it's been terrific to have this opportunity. I once in a while I've done talks like this in other countries. Okay, and so it's been great to be able to get involved in your group that is looking more in the Middle East. I've done talks in places like actually many talks in India, China, for other places in Asia, and so South America. I mean, there's a bunch of different people. I think this is really a good thing to do. Thank you, Fami, for doing this program, because I think that's what science is all about: is getting to know about different topics and getting to know people, and trying to figure out how it is that any individual can get involved in science in a given area, and make an impact. So anyway, so yeah, thank you very much. This has really been a
Thank
terrific
you.
opportunity.
Thank
Yeah,
you very much, dear Professor. Really appreciate your time, appreciate your presentation. I hope in future we can also see you again online,
yeah,
giving us your experience. Dear participants, this is the end of our spotlight. Thank you for all of you joining us, and thank you for Professor again for your nice presentation. Thank you, dear participant. We are sorry you couldn't answer all of your questions. You know, you have a time schedule behind. Thank you so much. Thank you again, and this is the end of our
thank you.
webinar. See you again.
Okay, bye now.